Wait for 5 minutes for other option.
हेलो हेलो। गुड मॉर्निंग उदय लक्ष्मी। हम वेट करेंगे कुछ 2-3 मिनट। गुड मॉर्निंग गुड मॉर्निंग। The two classes will be of each subject. Today class is on crop physiology and rest other two classes were on plant tissue culture. So crop physiology का भी 2 classes YouTube में होंगे, then rest everything will be on paid classes. पेड क्लासेस में ज़्यादा डिटेल में भी होगा और आपको अच्छे से गाइडेंस भी मिलेगी पेड क्लासेस में। आई थिंक और कोई नहीं आएंगे अभी, क्लासेस होंगे सबके मॉर्निंग में। सो ओके, स्टार्ट करते हैं हम। सो आज का टॉपिक रहेगा वाटर सस्पेंशन एंड कोलोइडल सॉल्यूशंस और कोलोइडल सिस्टम, और ये बेसिक है फॉर प्लांट वाटर रिलेशन। इट विल फॉर्म द फाउंडेशन टू अंडरस्टैंड प्लांट वाटर रिलेशन, basically the chemical nature of water or the structure of water and what is the physical importance of water. All these things we need to understand before entering into the main topic, that is plant and water relations, right? So why water is important? बताओ आप जो भी समझ रहे हो कि why water is important for plant. What are the role water play in plant system? Yes, it is essential for growth, but what are the exact roles that play by water which helps in growth and development of plant? There must be some roles, right, which promote or which enables the plant to survive. And we often heard that water is life, जल ही जीवन है. ओके, तो बताऊंगा ये, पहले देख लेते हैं वाटर। वाटर का फॉर्मूला सबको पता होगा, H2O, right? So water is made up of two hydrogen atom and one oxygen atom. Okay, and the structure is like this, V shape. The angle is 104.5 degree, but हम इसको बोल सकते हैं 105 degree also. एग्जाम में इतना ही आएगा, ये 104.5 it will not come. Okay, and it is, the structure is V shape structure. The bond angle is 105 degree and it is made up of covalent bond. So can anyone say what is covalent bond? कोवेलेंट बॉन्ड क्या होता है, आयनिक बॉन्ड क्या होते हैं? बॉन्ड हमेशा दो एलिमेंट के बीच ही फॉर्म होते हैं। तो शेयर्ड बॉन्ड मींस बॉन्ड इज शेयर्ड बाय टू एटम्स और इलेक्ट्रॉन इज शेयर्ड बाय टू एटम्स। कोवेलेंट बॉन्ड फॉर्म when two atom or two elements share their electrons with each other, and then they form a covalent bond. An ionic bond is formed due to attraction of opposite charges or ions with opposite charges. They attract each other to form ionic bond. It is the difference. Okay, so in water we see charge separation there, and due to the charge separation, the water molecule becomes a polar molecule. So what is charge separation? That forms due to the oxygen, electronegativity of oxygen. And what is electronegativity right now? So electronegativity is the tendency or ability of an atom in a chemical bond to pull the electron from its shared atom, right? So like in oxygen, in water, oxygen and hydrogen atom have formed bond. So oxygen is more electronegative. So it will pull the electron toward itself. Electron, it will pull toward itself. So due to this pulling or high electronegativity of oxygen atom, the oxygen will develop a partial negative charge, and as hydrogen is giving electrons to the oxygen, it will develop a partial positive charge, right? So due to this, in water molecule, they develop a polarity. So hence, the water molecule is a polar molecule. And why it is a polar molecule? Because of high electronegativity of oxygen, the uh, the charge separation occur between oxygen and hydrogen atom within a water molecule. That's why it become polar. Polar means it has two poles or some, there are poles. That's why it is called polar molecules. So here one is positive pole and another one negative pole. That's why water is considered as a polar molecule. Understood? Okay, on oxygen, partial negative charge develop, partial positive charge develop on hydrogen atom, and water is overall neutral. Okay, water as a whole means this molecule as a whole is neutral. The molecule as a whole doesn't have any charge or doesn't have any electrical charge. It is neutral as a whole, but internal polarity is there. Internal means within the molecule there is polarity. Okay, between hydrogen and oxygen atom. And because of band shape and charge separation this development of partial positive charge and partial negative charge, and band shape 105 degree. Because of this, one side of the molecule is slightly negative, slightly positive, and this water has a polar molecule. Because of the charge separation and development of this partial charge, water become polar. Okay, and now water also form hydrogen bond. Hydrogen bond has a lot of significance in the living organisms, and wherever the water takes part, the hydrogen bond play a crucial role in determining the physical properties or physical function of water, or whatever the function the water play, the hydrogen bond has a important role to play. Okay, so how the hydrogen bond form? Hydrogen bond form between two water molecules. It doesn't form between or within water molecule. Like covalent bond form within water molecule, bond form between water molecules. Means one water molecules will bond it with another water molecule by hydrogen bond. Okay, see here. This is one water molecule. This is another water molecule. And this is the third water molecule. Right? So here within the water molecule, this is covalent bond. And here, this is one water molecule, this is one water molecule. So this bond, this is hydrogen bond. Means hydrogen bond form between water molecules and covalent bond form within water molecule. So hydrogen bonds are intermolecular, and it is a weak electrostatic attraction. Why electrostatic attraction? Because in water molecule, in oxygen is developed partial negative charge, and water it is partial positive charge, right? Here also it is partial positive charge, here it is partial negative charge. Wait, partial positive charge, negative. So see, positive charge, negative charge, the charges attract each other to form this bond. That's why it is called electrostatic attraction. The hydrogen bond is formed due to electrostatic attraction between oppositely charged molecules or oppositely charged. But why it is not called ionic bond? Because the charge developed are partially developed, not completely developed charge. That's why they are not included in ionic bond, and they are called as hydrogen bond. Okay, and hydrogen forms between a hydrogen atom covalently bonded to an electronegative atom like oxygen or nitrogen. They are highly electronegative, and the most highly electronegative atom is fluorine. Sometimes it is asked, so highest electronegativity, fluorine has the highest electronegativity. Okay, and oxygen and nitrogen also they are electronegative, so they pull the electron from the atom with which they share their bond and become negatively charged or develop partial negative charge, and another electronegative atom O and end with a lone pair of electrons. See, this is an electronegative atom. So hydrogen atom attached to one electronegative atom form the bond, hydrogen bond with another electronegative atom, right? That is only here it has said. But hydrogen bonds are basically stronger than van der Waals and but weaker than ionic bond or covalent bond. Okay. So what are the physical properties of water and why physical properties of water is important? Why we should know the physical properties of water? What is the requirement to understand and know physical properties of water? You can say that, but this is not the answer I am looking for. To differentiate with other molecules, yes. After knowing, after knowing someone only you can differentiate. No, but what is the physiological importance within the plant or animals or anything? What is the physiological importance of knowing physical properties of water? Can we say if अगर हमें physical properties पता चल जाएगा तो how water will behave inside a biological system? Can we predict that? Can we know the importance of water in a biological system if we understand the physical properties of water? So physical properties, not only water, if you know the physical properties of any substance, it will be helpful to understand them better in the biological system, right? So pure water is colorless, odorless, tasteless. This we are like learning or reading from class 6 or class 7th, right? So nothing is there, and it is transparent in small amounts and pale blue in large volume. Why in large volume water looks blue? ये वही सेम कॉन्सेप्ट है जिसकी वजह से स्काई ब्लू दिखता है. Why sky looks blue? There is a reason behind it, and the same reason is behind why water in large amount also looks blue. No, 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 no. It's not about density. It has something to do with the light. The physics wala part it is. Okay, because of scattering of light, or scattering of blue light. Blue light has the like the smallest wavelength, so it scattered more. So because the water in large quantity scatter the blue light more, it looks blue. In the sky also it is same. The blue light scatter much more. That's why sky also looks blue. Not reflection, it is refraction. Okay, so boiling point of water is 100 degrees Celsius. At what atmospheric temperature? And melting point is 0 degrees Celsius of water. But these are very much more. If we see the size of water molecules as compared to its size, the melting and boiling point is very high. And the reason being hydrogen bond. Because of hydrogen bonding, the boiling point and melting point of water are very high expected from a molecule of its size. The water molecule has very small size, so it should have low boiling point and low melting point. But because of hydrogen bonding, the melting point and boiling point of water are very high. Okay, if you see the other molecules of size of water molecule, they have low melting point and boiling point than water because they lack hydrogen bonding. So it is the importance of hydrogen bonding. Because of hydrogen bonding, only water is so much essential, or so much water has so much to do in the living system. Okay, and maximum density is 4 degrees Celsius. That is 1 gram per centimeter cube. Below 4 degrees Celsius, water expand instead of contracting. Why? Why? Generally, what happens when you cool a system, the volume decreases and the density increases. Like it get contracted. If you cool a system or cool something, the volume will decrease, it will contract, and the density increase. But in case of water, the density increases until 4 degrees Celsius. And below 4 degrees Celsius, the volume increases, means the water expand, and the density decreases below 4 degrees Celsius. Why? Okay, so below 4 degrees Celsius, what happened? Up to 4 degrees Celsius, the contraction occurred, but below 4 degrees Celsius, what happened? The hydrogen bond become very stronger because the water molecules get closer and closer up to a point, and when at 4 degrees Celsius they become so much closer that they cannot contract much, and up below 4 degrees Celsius the hydrogen bond becomes stronger, and it form a open lattice structure, means the hydrogen form a hexagonal structure and fixed at the lattice. Lattice at this point are lattice. Okay, and it expand in volume. So if the water molecule expand, the volume will increase, and when the volume will increase, the density decrease. That is the reason why ice float on the water, because ice has less density than water, and the substance with lesser density will float on the substance of higher density. That is the basic reason. Okay, and why it occurs below 4 degrees Celsius? Why the volume of water increases? Because of hydrogen bonding. Okay, what is specific heat capacity? Do you know? What is specific heat capacity? See, amount of heat required to raise the temperature of 1 unit of substance by 1 degree Celsius. Okay, So how much quantity of heat is required to increase the temperature by 1 degree Celsius of 1 unit of any substance? So for example, take water, and the temperature right now, I'm at Delhi, so temperature right now would be around 15 to I think 20 degrees Celsius right now, around 20 degrees Celsius. So let's say water is also on 20 degrees Celsius, but we want to increase the temperature from 20 degrees Celsius to 21 degrees Celsius. Okay, it is 1 degree Celsius increase. So how much heat I should provide to the water so that the temperature will change to 21 degrees Celsius from 20 degrees Celsius. So the amount of heat I have provided to water to increase its temperature by 1 degree Celsius will be the specific heat capacity of water. Okay. So that is specific heat capacity, and water has very high specific heat capacity, and that is also due to the hydrogen bonding, because a lot more heat is required to break the hydrogen bonding and increase its temperature by 1 degree Celsius. If hydrogen bonding would not have been there in the water molecule, the specific heat capacity of water would have been less, much less. So because of hydrogen bonding, the specific heat capacity of water become high, that is 4.18 joule per gram per degree Celsius. Got it? Remember this. Sometimes it is asked in examination that what is the specific heat capacity of water. It is per gram. This unit is important. Gram and degree Celsius. So remember the unit as well. Joule, gram per degree Celsius. Okay. So what is the meaning? It takes a lot of energy to change water's temperature due to hydrogen bond. Hydrogen bond is the reason because of which the water need more heat to increase the temperature by 1 degree Celsius. Okay, what is the biological importance then? If specific heat capacity is more, what it can be like, what will be the benefit for living system? Okay, the biological importance would be regulation of temperature, right, within the body. As a human or as an animal, we are exposed to different temperatures, right? In morning it will be cold, afternoon will be under sun, evening again cold. So there are changing temperature. So how water help in regulating the temperature changes in our body? And as we know, the enzymes require a specific range of temperature to function, and our whole system or living system are run by enzymes or chemicals, and all the reactions that occur in our body require a specific temperature, right? So water helps in maintaining that temperature, but how? Because of high heat specific capacity, because a lot more heat is required to change the temperature of the water body, right? And our body is composed of around 80% of water, I guess 70 to 80% of our body is of water. So that is the reason why our enzymes or entire system is functioning, because water helps in maintaining a particular range of temperature where the enzymes and all other reaction takes place, and we continue to live. Okay, so living organism, soil and atmosphere also. Evaporative cooling, when we apply water, it absorb the like the water absorb the heat and get evaporated, so it maintains the temperature of soil as well and atmosphere as well, and protects plants and animals both from sudden temperature changes. Like it is very difficult to change the temperature of water, right? Because the specific heat capacity, it absorb more heat to change its temperature by 1 degree Celsius. So more heat is required to change its temperature. So that is the reason it prevent the sudden temperature changes. It take a lot of time to change the temperature, and that is how water helps in maintaining the temperature or regulate the temperature of living systems. And another property, that is cohesion and adhesion. Cohesion, the word co means similar. So it is the attraction between similar molecules. So it is the attraction between water and water molecules. Attraction between water and water molecules. And adhesion is attraction between water and other surfaces. It may be the glasses or the bank of a river, anything. Okay, example, water forming droplets. That is your surface tension. Okay, water sticking to glass. And because of this cohesion and adhesion property, the water or sap, they are move upward or in the xylem, through xylem water moves in the plant. So because of the cohesion and adhesion property only, the water can travel or move inside the xylem or inside the plant because of the cohesion and adhesion property. And another one is there also, surface tension. Surface tension is due to cohesion only. Cohesion is the reason for surface tension, and surface tension help in reducing the surface area of the water, and also it develop a tension so that the transpiration pool can occur. Okay. See, next, surface tension caused by cohesion at the air water interface. So for example, think this is a beaker. Okay, and this is the water surface. Here water molecule would be here. Here are the water molecules. So here in water molecules, there are cohesion, attraction between both due to hydrogen bonding only. Because of hydrogen bonding, there will be attraction here, here, here. But the water molecule at the surface do not experience any force from upside. Like there is no water molecule to attract at the upper side. So what will happen on this water molecule? The only force was from inside. So they are वो नीचे ही खींचते हैं. So the water molecule at the surface are only pulled downward. There is no upward cancellation. Okay, so what happened? So here the water surface will form like this. You might have noticed in any glass the water surface like this. But why here they remain like this? Because of adhesion. Adhesion. Adhesion is the attraction between water molecule and some other atom molecule or some other substance molecule. That here it is glass. That is surface tension. Due to surface tension, the water molecule pulled inward in a glass and create tension. And this tension is known as surface tension. Okay, what is the importance of surface tension? The water has high surface tension, enables transpiration pool in the plant, water to form droplets. Droplets, why they form droplets? Because surface tension caused them to reduce their available surface area. So because of surface tension, they minimize their surface area and form droplet. So surface tension is the reason why water form droplet and the transpiration pool occur, and insect work on water. Because of the tension developed due to the attraction or cohesion between the water molecules, surface tension develop, and because of the surface tension, insect can walk on the water surface, because the tension or the force applied by the insect is less than the surface tension. So if someone to dive into the water, they have to break the surface tension, or they have to apply the force more than surface tension. But insect cannot apply the force more than surface tension. That's why they walk on the water surface. आप लोगों ने भी एक्सपीरियंस किया होगा ना कि जब ज़्यादा ऊंचाई से जब कूदते हैं हम पानी में, दर्द होता है. We get pain. The water splash, it cause pain to us, right? That is due to surface tension, because the surface tension force and our force, they both collide, and that is why it like impact us. So water is high tensile strength. They can be pulled without breaking. Okay. Why water is strong? Cohesion from hydrogen bonding. That's why it is strong. We have already seen. They resist the pulling force without breaking because of hydrogen bonding only. And that's why it became important for biological role. Because of this property, that high pulling force, the tensile and hydrogen bonding, cohesion, adhesion, surface tension, because of these properties, the physical properties of water make it very essential. Okay, and also polar. Water is polar, so that it can be. That is the reason why water is a very good solvent, right? If water would not have been polar, it cannot or dissolve the substance or cannot become the good solvent. Water is important in living organisms because it is a good solvent as well. Good solvent. Solvent is a dispersion medium on which the solutes are getting dispersed or getting mixed. Okay. Pull upward in xylem to tall trees. Essential for water transport in plant. Okay. The properties of water makes it very good to play a great role in biological system. See, universal solvent. Highly polar. That's why it is an excellent solvent. Dissolve ions, sugars, proteins. How? Polar water molecules surround the solute particles. See, this is a solute particles, maybe NaCl. Let's see. Okay, this polar water molecule, your polar water molecule will surround the solute, and we know this has oxygen as negative and hydrogen has partial positive charge, right? So NaCl, positive and negative. This charge are pulled. Na will be pulled by oxygen part and Cl will be, so in water they will become Na plus and Cl negative. And if the water would not have been polar, they cannot dissociate, they will not be able to dissociate Na and Cl into Na plus and Cl negative. Okay, because of polarity they can dissolve the ionic compounds also. First they surround the solute particles, then electrostatic interactions break solute bonds, dissolve them. This is only electrostatic attraction, that Na plus is attracted to O negative and Cl minus is attracted to H plus. Okay, water dissolves more substance than any other liquid on earth. Water is the only liquid which dissolves the almost all solids. Almost all. Not all, but almost all. There are some exceptions which are not dissolved by water. They are dissolved in fat. So water contains 80 to 95% of growing plant tissue contain 80 to 95% of water. Means the tissue has 80 to 95% of water of their total mass. So even dry tissue have 5 to 15% of water. Means they are called dry tissue, but they also contain water. Okay, universal substance we have solvent we have seen that salt, sugar, amino acid, hormones. It allows molecule to move freely inside the cell. Yes, water and protoplasm function. Water is also important for water and protoplasm function. Protoplasm is a living part of the cell. Means if it is plant cell, see if it is plant cell, this is called protoplasm. This is cell wall. This is protoplasm. Okay. And water keeps the protoplasm fluid and active. The protoplasm to function, it must be in fluid state. The cytoplasm inside the protoplasm, it must be in fluid state to have a proper function, because the proteins, fats and other organelles are dissolved, or sorry, are suspended in the cytoplasm only. So for the cytoplasmic streaming, it should be in liquid state. And what provide the cytoplasm to be in liquid state? It is the water. Because of water, the cytoplasm are in liquid state so that it can perform its function of cytoplasmic streaming, or the suspended particles in the cytoplasm do their work or able to perform their assigned functions. We hear water influence macromolecules also. How water influence protein? Maintain the shape and function of protein in the cytoplasm or in any organelle. Nucleic acid also, replication and transcription. Water help. Polysaccharides aids in synthesis and breakdown. They break down hydrolysis, synthesis, hydrolysis, hydration, dehydration. These are the processes which require water. Hydrolysis is breaking the molecule. Hydration is adding water to molecule, means adsorption, so that it can absorb water and then perform its function. Dehydration, removing water. Water is not just a medium, it's a reactant too. Reactant means there are two things, no? Reactant plus it will give a product. Reactant side, product side. So water is also act as reactant. Can you remember one reaction where the water is act as reactant? Photosynthesis. Are you heard of? Are you aware of photosynthesis? In photosynthesis, water act as a reactant and they produce sugar and oxygen. So water is important to provide electrons that is required in photosynthesis, and also hydrogen for making glucose, and also it is water is the molecule which is also responsible for release of oxygen. Okay, in photosynthesis, there is a special class on photosynthesis only, not only one class, there are a lot of class in photosynthesis. Okay, so there we will see in very detail how how oxygen is released from water, and how the scientists confirm that water is the molecule or water is the substance which is responsible for release of oxygen from plant by the process of photosynthesis. Okay, without it, no photosynthesis. If water is absent, photosynthesis cannot occur. And photosynthesis is the base of all food. There would be no food except photosynthesis. Like in tissue culture class, we have seen that plants are the base of all food chains. Like the everything or all the energy we derive from the plants. That's why plants are called the producer, right? If photosynthesis would not have been there, the plant also would have been unable to produce the food. प्लांट भी खाना नहीं बना पाते ना, तो प्लांट ही खाना नहीं बना पाते तो हमको कहाँ से खाना मिलता? So water is that much important for all the living things. That's why we say जल ही जीवन है. Water regular temperature हमने देखा ये, we have already seen. And water के turgor pressure. What is turgor pressure? What is turgor pressure? 5 students live देख रहे हो. Only Uday Lakshmi is interacting with me. बाकी भी करो इंटरेक्ट, कोई नहीं, गलत होगा क्या हो जाएगा? गलती सबसे होती है, इंसान गलती का पुतला है. ट्राई टू आंसर. टर्गर प्रेशर क्या होता है? यूजी में पढ़ा होगा ना आपने. You might have studied in under graduation. Yes, pressure developed against the cell wall. Only Uday Lakshmi is answering. There are 5 students who are currently viewing. So suppose this is cell. So when water enters into the cell, it will become full. and because of the entering water, it exerts pressure against the cell wall. The pressure exerted by water against the cell wall is known as turgor pressure. Internal pressure against the cell wall. Okay. What is the function of turgor pressure? Cell enlargement, stomatal movement. Yes. When the guard cell absorb water or the water enters to the guard cell, it become the turgor pressure helps to open the stomatal pore so that the gas can exchange through the stomatal pore. So in stomatal movement, we will see this in following classes. The stomatal movement, water helps. Phloem transport, membrane transport, safe maintenance, shielding emergence. In almost in every function of plant, water is essential. Almost in every function. Okay. And for so agriculture, we know if the water is essential for physiology of plant or in all the physiological process of plant. Agriculture is also based on plants only, no? So for agriculture also water would be important. Like germination, growth rate, nutrient uptake and yield. These are the importance of water. So this is summary. Water in plants, physiological role. Trace tissue, solvent for all molecules, biochemical hydration, photosynthesis, physiological load, temperature, pressure, transpiration, transport, agriculture load, productivity के लिए, and for life. Right. So now let's come to solution. What is solution? बस डेफिनेशन देखेंगे और कुछ नहीं है उसमें ज़्यादा इंपॉर्टेंट. और कौन सा सॉल्यूशन स्टेबल होता है, स्टेबल कब बोला जाता है सॉल्यूशन को, वो देखेंगे. Solution is a homogeneous mixture of more than one substance, right? It may be like, for example, salt in water, sugar in water, homogeneous. Why it is called homogeneous? Because it is very difficult to identify the solute or to see the solute in the solution, right? You cannot see the solute in the solution. That is why it is homogeneous. Complete mix. पूरा मिक्स हो जाते हैं और हम सोल्यूट को आइडेंटिफाई नहीं कर पाते हैं. इवन अंडर द माइक्रोस्कोप वी कैन नॉट सी द सोल्यूट. दैट्स व्हाई इट इज कॉल्ड होमोजीनस. पूरा मिक्स हो जाता है इसीलिए यूनिफॉर्म और या होमोजीनस कहा जाता है. और स्टेबल क्यों कहा जाता है? स्टेबल इसीलिए कि वो सॉल्यूशन जिस स्टेट में है, मतलब मिक्स्ड स्टेट में है, उसमें कुछ दिखाई नहीं दे रहा है, बस वाटर जैसा लग रहा है लेकिन सॉल्ट है उसमें, तो उसको ऐसे रख भी देंगे हम लोग. इफ वी कीप इट स्टेडी, the solute particles will not settle down at the bottom of the container. That's why it is stable. Means जैसा है वैसा ही रहता है solution. But suspension में क्या होता है? Suspension is the heterogeneous mixture and also called unstable. Why unstable? Because if we keep the suspension steady, slowly the solute particles will settle down and form a clump at the bottom of the container. Right? That's why that is called unstable and heterogeneous. Yes, you cannot separate, at least through ordinary filter paper, but there are some methods through which we can separate the solution also homogeneous mixture also, but not usually through ordinary filter paper or ordinary filtration technique. We cannot separate. Right. So what are solute and what is solute and what is solvent? The solute is the dispersed medium, right? Means the thing which is dispersed into a solvent, that is generally in smaller quantity that present. Okay, and the solvent are present in larger quantity. For example, sugar, salt, these are solute, and water is a solvent. Dispersion medium and dispersed phase. Dispersion. Okay. So why it is stable? Solute particles spread evenly throughout the solvent. They do not settle down. If something is not settling down and it is as it is after and before keeping it steady, जैसा था वैसा ही रहेगा तो स्टेबल है. और अगर चेंज हो रहा है, if it is changing and the solute particles are getting settled down, then it is unstable as it is in suspension, right? So here are some types of solution like solid, liquid, salt in water. The examples are there. You can see it, go through it. Crystal wet, not important. let it be. So suspension, heterogeneous, solid and liquid. Okay, and where is unstable? It is unstable. Unstable. They settle down after some time. That's why unstable. Clear water remains above and solid settles below. Particles are visible to naked eye under microscope. Can't be filtered. Can be filtered easily. Suspensions cannot pass through parchment or ion membrane. No diffusion, no osmosis. Suspension cannot participate in diffusion or osmosis. Okay. So the dispersion is the medium. Solvent is dispersion on which the solute particles are dispersed. Solute particles get dispersed, no? You know the meaning of disperse? Scattered or spreaded. So dispersed is what get dispersed in the medium, and dispersion is the water or the solvent. Dispersion medium and dispersed phase. Understood, Udalakshmi? Okay, okay. So here, difference between solution and suspension. Homogeneous, heterogeneous, very small particles. Like it is solute. Solute is very small and large. Visibility, invisible, can be visible. Stability, stable, is unstable because settle down. Filtration cannot be filtered, can be filtered. Membrane passes through the membrane, does not pass. Example. Okay. Now colloids and colloidal system. So we saw solution, we saw suspension. So in between these two, there one more thing lies, that is called colloids or colloidal solutions. It is on the basis of size of solute particles. Okay. So colloids are intermediate between solution and suspension. It is the beej ka wala. Okay. Because of its size. The colloids are intermediate between solution and suspension. And the colloids are named by Thomas Graham. He is the person who named the particle size which lies between solution and suspension as colloids. Okay. So colloid is a heterogeneous system but stable. Colloids are heterogeneous, means the solute particles and the solvent cannot mix. Okay, but they are stable. The solute particles do not settle down when it is kept steady. See what was there in solution? It is homogeneous and also stable. But in suspension, it was heterogeneous and unstable. But what happened in colloid? It is heterogeneous but stable. Okay. So for example, clay in water. All the soils are not clay. Clay is like very small size. So if you dissolve only clay, no, it will act like colloids. It will not settle down. Don't settle or dissolve. It will neither dissolve or not settle. Solution, it is completely dissolved. Suspension, not dissolved. And in suspension, it was settling down. Solution, not settling down. But in clay, in colloids, it will neither settle or neither dissolve. It is intermediate between solution and suspension. So particle size comparison, suspension 10 to the minus 5 to 10 to the minus 6 centimeter, colloid between suspension and solution, and here it is minus 5, that's why it is larger. The size of particles is larger in suspension and smallest in true solutions, and colloid, the intermediate between the two. Visible to next, stability, unstable, it is stable, very stable. Okay. So see here, dispersed phase, dispersion medium, lyophilic colloid, lyophobic colloid. Philic means loving, lyo means solvent. Lyo, solvent, phobic means heating. The solvent heating, solvent loving. Dispersion medium, the substance present in larger quantity is dispersion medium on which the solutes are getting dispersed. Dispersed phase, the substance present in smaller quantity, means solute. Solute are known as dispersed phase and the solvent are known as dispersion medium. Examples are there, so no need. The types of colloidal system. If dispersed phase is solid and dispersion medium is liquid, the type of colloidal solution which is known as sol. It is sometimes asked in examination. If liquid will be there, it is called emulsion, like oil in water or milk, cream. There is liquid in liquid. Gas in liquid, foam. Solid in gas, smoke. And liquid in gas, mist. Like example, fog or cloud, that is smoke from fire. These are the examples. This sometimes asked in examination. So remember this table. And it is very easy. There is nothing. Lyophilic versus lyophobic. Lyo means solvent. Philic, love. Phobic, hate. Okay, affinity for solvent. Lyophilic has high affinity for solvent. Lyophobic has low affinity for solvent. Stability, very stable because it has high affinity for solvent, means it will form a good bond with the solvent. So it will mix well. So if it is mixed well, it will be stable, right? So that's why lyophilic are more stable and lyophobic are less stable. Reversibility, yes, it is reversible, but it is irreversible. We will see this. Why? Why lyophilic colloids are reversible, but it is irreversible? Have you seen the agarose gel? If we heat this, it will convert it into liquid, and again, it will convert it back to solid, right? But in egg albumin, after heating, it will become solid, but it cannot be reversed back because in egg, the attraction between them is very less, so they were unstable. Preparation is very easy, just mix. Require special methods. Starch and gelatin example are there. Sol gel transformation. So it is called gelation. Sol to gel, semi-solid. Sol is liquid, right? Solid in liquid, so it is in liquid phase. So gelation is a process that convert the sol stage or sol phase to gel phase. Gel phase is semi-solid, triggered by cooling, chemical change or concentration. Generally by cooling. If you cool the sol or gel phase, it will become gel. And solation means by heating. Heating or dilution, you can do it. That gel or semi-solid part converted into sol. It is liquid. Reversible versus irreversible colloids can switch between sol and gel. That agarose I told you, no, that can switch between sol, that is in liquid phase, and gel phase, semi-solid phase, like agar. Okay, and irreversible colloid, they cannot switch between sol and gel. Gel is the semi-solid one. It is also, gel is basically liquid in solid. Solid is the dispersion medium here, and liquid is the dispersed phase here. But in sol, what happened? It is solid in liquid. Solid in liquid. Properties of colloidal solution. Yes, filterability. We can filter, but not with ordinary filter paper. We have to use like ultra filters like parchment paper or colloidal membrane. These two membrane, remember, with the help of which we can filter out the colloidal solution, colloidal particles. Okay, but they are small enough to pass through ordinary filter papers. So colloidal particles can pass through ordinary filter paper, but they can be filter out or they can be separated with the help of parchment and colloidal membranes. Okay, so they are in between the true solution and suspension. We have already seen that. And adsorption and surface area. Colloidal particles have high surface area. We know the smaller the particles, more the surface area, right? So colloidal solution has more surface area than the particles in suspension, but has less surface area than the true solution. True solution में तो exist ही नहीं करता solute, तो इसलिए उसको count करना नहीं है. और सस्पेंशन में बहुत बड़े हो जाते हैं, तो उनका सरफेस एरिया कम होता है। एंड फॉर रिएक्शन टू अकर वी नीड द मोर सरफेस एरिया। अगर सरफेस एरिया ज़्यादा होगा तो रिएक्शन भी अच्छे से हो पाएगा। दैट्स वाई कोलोइडल पार्टिकल्स आर गुड फॉर रिएक्शन। और हमारा जो सॉइल सेल में जो साइटोप्लाज्म होता है वो कोलोइडल स्टेट में रहता है। द साइटोप्लाज्म inside our cell or living system are in colloidal system, like in colloidal form, or colloidal solution. Okay, so colloidal particles are good in adsorption and has high surface area, have large huge surface area because they're tiny and numerous. बहुत सारे होते हैं और छोटे छोटे होते हैं. They are adsorb ions molecules on their surface. कोलोइडल पार्टिकल्स का ना चार्ज डेवलप होता है उनके ऊपर, दैट्स वाई वो एड्जॉर्ब कर पाते हैं चीज़ों को. सॉइल साइंस में आपने पढ़ा होगा सॉइल कोलोइड्स. सॉइल कोलोइड्स आर रेस्पॉन्सिबल फॉर कैटायन एंड एनायन एक्सचेंज. सेम कोलोइडल वही रहता है. कोलोइडल पार्टिकल्स में चार्ज डेवलप होते हैं, चार्ज रहते हैं, दे आर चार्ज पार्टिकल्स, so they help in adsorption. Adsorption is the reason colloids are important in biochemical reaction inside the protoplasm. Okay, and also colloidal systems show Tyndall effect. What is Tyndall effect? The colloidal particles are large enough to scatter the light. So when light pass through a colloidal solution, it cannot. सॉल्यूशन में क्या होता है, ट्रांसपेरेंट होते हैं, तो लाइट पास हो सकता है एक साइड से दूसरे साइड तक. बट कोलोइडल सिस्टम में क्या होता है, जब लाइट एंटर करता है, व्हेन लाइट एंटर द कोलोइडल सिस्टम, द पार्टिकल्स इनसाइड द कोलोइड सिस्टम स्कैटर द लाइट एंड इट डज नॉट अलाउ द लाइट टू पास थ्रू इट. Right, it entrap the light inside the solution only. Yes, scattering of light particles because of the colloidal particles, because they are larger than solution, and because of that it create effect that लगता है कि वो colloidal system ही glow कर रहा है. तो उसको जो एक टिंडल करके एक साइंटिस्ट था, उसने उसको मतलब डिस्कवर किया था, तो इसलिए उसके नाम से जाना जाता है टिंडल इफेक्ट. So when a strong light beam passes through colloids, the path of light become visible. This happens due to scattering of light by colloidal particles, more visible in lyophobic soils than lyophilic soils. Lyophobic में ज़्यादा क्यों दिखाई देता है, क्योंकि lyophobic में सॉल्वेंट हिटिंग, तो अच्छे से मिक्स नहीं हो पाएंगे, तो कोलोइडल पार्टिकल्स जो होंगे वो अच्छे से विजिबल होंगे. राइट, तो जितना ज़्यादा पार्टिकल रहेगा वो उतना अच्छे से स्कैटर करेंगे, और जितने अच्छे से अगर मिक्स हो गए तो अच्छे से नहीं कर पाएंगे स्कैटर. सिंपल लॉजिक है. ओके, अल्ट्रा माइक्रोस्कोप ऑब्जर्वेशन. अल्ट्रा माइक्रोस्कोप में यस ब्राइट स्पॉट दिखते हैं ऑन ए डार्क बैकग्राउंड, मतलब ऐसे एक डार्क बैकग्राउंड है, इसमें ब्राइट स्पॉट दिखेंगे कोलोइडल पार्टिकल्स, अगर माइक्रोस्कोप के अंडर आप उसको ऑब्जर्व करोगे तो. These are not the particles themselves but halos of scattered light. ये पार्टिकल खुद नहीं होते, जो लाइट स्कैटर होता है वो दिखता है डॉट 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 डॉट. ओके, दे आर स्कैटरिंग द लाइट, तो उसी से दिखता है. ओके, एंड ब्राउनियन मूवमेंट बेसिकली जिगजैग मूवमेंट क्यों होता है, बिकॉज ऑफ अनइक्वल डिस्ट्रीब्यूशन ऑफ चार्ज और अनइक्वल बॉम्बार्डमेंट. ठीक है, मतलब पार्टिकल्स रहते हैं और पार्टिकल्स का साइज तो डिफरेंट होता है, दे कोलाइड विथ ईच अदर एंड द कोलाइजन रिजल्ट इन वेरियस मूवमेंट, एंड दिस मूवमेंट इंक्रीजेस विथ इंक्रीजेस टेम्परेचर. और जैसे ही पार्टिकल साइज बढ़ता है तो ये मूवमेंट डिक्रीज हो जाता है, क्योंकि जो लार्ज पार्टिकल्स होते हैं वो कोलाइड होने के बाद उस तरीके से मूव नहीं कर सकते, उसी स्पीड से मूव नहीं कर सकते. ठीक है, और विस्कोसिटी बढ़ गया. विस्कोसिटी इज अ रेजिस्टेंस टू फ्लो. राइट, मीन्स हायर द विस्कोसिटी it will more resistance to flow. So if viscosity will increase, the Brownian movement will decrease. And if we want to increase the Brownian movement, we have to decrease the viscosity of the liquid, right? And smaller the particle size, higher will be the movement. Higher is the temperature, higher will be the movement. This is basically more vigorous. Helps in prevent settling. Because of Browning movement, the colloids in particles, the colloidal particles do not settle down because of this movement or अनइक्वल बॉम्बार्डमेंट ऑफ सॉल्वेंट मॉलिक्यूल्स। तो सॉल्वेंट मॉलिक्यूल्स होते हैं वो एक दूसरे के साथ अनइक्वली कोलाइड करते हैं, दैट इज द रीजन व्हाई कोलॉइड्स आर स्टेबल। Also it is electric property because colloidal particles carry a uniform electric charge, all positive और all negative। जो colloidal particles होते हैं उनके पास uniform negative charges होते हैं या positive charges होंगे, equal ही होते हैं। So that's why equal charge repel each other, we know that, that is why they cause Brownian movement, that is also one of the reason। bombardment तो होता है पर इक्वल चार्ज होते हैं तो एक दूसरे को रिपेल भी करते हैं अंदर, तो रिपेल करने की वजह से वो मूवमेंट एक्वायर करते हैं, और मूवमेंट एक्वायर करते हैं तो एक दूसरे के साथ कोलाइड भी करते हैं। एंड चार्ज कैसे आता है? बिकॉज ऑफ एड्जॉर्प्शन ऑफ आयन। उनके पास जो हायर सरफेस एरिया रहता है, कोलॉइड हैव हायर ह्यूज सरफेस एरिया, तो सरफेस एरिया हाई होने की वजह से दे एड्जॉर्ब द मॉलिक्यूल्स, एंड बिकॉज ऑफ एड्जॉर्प्शन दे डेराइव चार्जेस, and because of charges they either repel each other or attract each other, and because of repulsion and attraction they move here and there and collide with each other, and because of the collision Brownian movement occur। And because of the Brownian movement the colloidal solution is stable, it does not settle down। Okay, electrophoresis। Oh, one more thing is there, coagulation। coagulation flocculation, कोआगुलेशन और फ्लोकुलेशन का मतलब होता है कि एक साथ क्लम्प होके, मतलब एक दूसरे से जुड़ जाना, अटैच हो जाना, वही होता है कोआगुलेशन फ्लोकुलेशन। एक फ्लॉक बनाना या कोआगुलेट हो जाना। फ्लॉक मतलब जो क्लम्प एक साथ जुड़ जाते हैं वही होता है। तो कोआगुलेशन फ्लोकुलेशन होता है कोलोइडल पार्टिकल्स में कब? जब इलेक्ट्रोलाइट ऐड किया जाता है। इलेक्ट्रोलाइट्स आर बेसिकली सॉल्ट्स, तो अगर सॉल्ट ऐड कर दोगे तो जो अपोजिटली चार्ज पार्टिकल्स होंगे दे अट्रैक्ट ईच अदर एंड दे फॉर्म बॉन्ड, और बॉन्ड फॉर्म करने के बाद वो सेटल डाउन हो जाते हैं धीरे धीरे। सो एडिशन ऑफ इलेक्ट्रोलाइट न्यूट्रलाइजेस द चार्ज। जो कोलोइडल पार्टिकल्स में चार्ज होते हैं ना, चार्ज पार्टिकल्स, वो जाके ऐड हो गए, उसमें रिएक्शन करके बॉन्ड बना लिया, आयोनिक बॉन्ड बना लेते हैं, तो वो क्या होता है, न्यूट्रलाइज कर देते हैं सॉल्यूशन के चार्ज को। अगर एक बार न्यूट्रलाइज हो गया तो रिपल्सन और अट्रैक्शन खत्म हो जाएगा, तो उसके लिए ब्राउनियन मूवमेंट भी खत्म हो जाएगा, कोलाइजन ही नहीं होगा। अगर और मूवमेंट खत्म हो गया तो दे विल सेटल डाउन। So addition of electrolytes into the colloidal solution help in coagulation or flocculation। Okay, without repulsion particles clump together and settle। Repulsion क्यों नहीं होगा? Because the addition of electrolyte neutralize the charges। Okay, mutual coagulation means mixing positive and negative charges neutralize each other। तो जो पॉजिटिव और नेगेटिव चार्जेस होंगे इलेक्ट्रोलाइट अगर आप ऐड करोगे तो वो सेटल डाउन हो जाएंगे। ऑस्मोटिक प्रेशर, बहुत लो ऑस्मोटिक प्रेशर रहता है कंपेयर टू ट्रू सॉल्यूशन। ओके, व्हाई वी विल सी, because particles number is small through the size is larger। पार्टिकल के नंबर कम होते हैं कोलोइडल सॉल्यूशन में, because the solute particles, number of solute particles is less in colloidal solution because the size is larger as compared to true solution। okay, it is relative concept। तो रिलेटिव कॉन्सेप्ट में क्या होता है, सॉल्यूशन में जितने सोल्यूट पार्टिकल्स रहते हैं, नंबर ऑफ सोल्यूट पार्टिकल्स कोलोइडल सॉल्यूशन में उससे कम रहते हैं, लेस रहते हैं, ठीक है। एंड द ऑस्मोटिक प्रेशर इज डिपेंड ऑन द नंबर ऑफ सोल्यूट पार्टिकल्स प्रेजेंट, और द concentration is calculated on the basis of number, right? So the particle number is small in colloidal solution as compared to true solution, that's why the colloidal solution has low osmotic pressure as compared to true solution। the difference between colloid suspension ये देख लेना, समझ में ना आए तो बता, पूछ सकते हो मुझे, पर ये सब मैंने एक्सप्लेन तो कर दिया, आपको समझ में आ जाएगा। प्रोटोप्लाज्म का कोलोइडल नेचर रहता है, ये भी बताया था। प्रोटोप्लाज्म जो सेल वॉल को छोड़ के प्लाज्मा में उनके अंदर जो भी रहता है उसको प्रोटोप्लाज्म बोलते हैं। साइटोप्लाज्म प्लस न्यूक्लियस इक्वल टू प्रोटोप्लाज्म। ओके, मोस्टली वाटर 70 to 90% we have already see। लिविंग सब्सटेंस इज इनसाइड सेल, साइटोप्लाज्म प्लस न्यूक्लियस इज प्रोटोप्लाज्म। इट बिहेव्स लाइक कॉम्प्लेक्स कोलोइडल सिस्टम, दैट्स आल्सो आई हैव ऑलरेडी टोल्ड। लार्ज प्रोटीन मॉलिक्यूल्स सस्पेंडेड रहते हैं वहाँ पे, एंड अदर मैक्रोमोलिक्यूल्स लाइक कार्बोहाइड्रेट, फैट्स, प्रोटीन्स, ये सब they remain dispersed in water forming a colloidal sol or gel system, already seen। dynamic nature, protoplasm can shift between sol and gel, ये भी बताया, depending on conditions। साइटोप्लाज्म स्ट्रीमिंग होता है, ये भी मैंने बताया है आपको। ओके, की प्रोटोप्लाज्म का प्रॉपर्टी क्या होता है एज ए कोलोइड? कोलोइडल के प्रॉपर्टीज क्या क्या थे? हेट्रोजीनस था बट स्टेबल था, राइट। रिवर्स हो सकते थे, जो मतलब सॉल जो है दैट इज सॉलिड इन लिक्विड, और जेल दैट इज लिक्विड इन सॉलिड, रिवर्स हो सकते थे, एक्सचेंज कर सकते हैं एक दूसरे में, फ्लेक्सिबिलिटी है, सेलेक्टिविटीज, राइट। और क्या है, कोलोइडल पार्टिकल्स का लार्ज सरफेस एरिया था। तो हमें प्रोटोप्लाज्म में क्या है, जो प्रोटीन होता है दैट इज ऑल्सो प्रेजेंट इन कोलोइडल स्टेट। तो प्रोटीन हैज हाई सरफेस एरिया एंड एंजाइम्स आर बेसिकली प्रोटीन ओनली, राइट। सो दे एंजाइम में होता है ना, एड्जॉर्ब करते हैं सबस्ट्रेट को, फिर एक रिएक्शन कैटलाइज करते हैं, राइट। एंड ऑल्सो कोलोइडल पार्टिकल्स आर डायनामिक, जैसे इलेक्ट्रोलाइट हमने डाले कोआगुलेट हो गए, फिर हम उसको फिर से अगर शेक कर देंगे तो फिर वो मिक्स हो सकते हैं, तो डायनामिक रहते हैं, अडेप्ट टू मेटाबॉलिक नीड्स। ओके, सो आज का लेक्चर इतना ही था, और हम फिर मिलेंगे शायद 3 और 4 अराउंड, वी विल अगेन मीट फॉर द नेक्स्ट पार्ट ऑफ प्लांट वाटर, द फंडामेंटल पार्ट ऑफ प्लांट वाटर रिलेशन। हम उसमें शायद डिफ्यूजन ऑस्मोसिस ये देखेंगे आज। अभी 3 और 4 PM क्लास विल बी देयर, लिंक शेयर हो जाएगा आपको। तो हैव यू अंडरस्टूड वॉट एवर आई हैव एक्सप्लेन टूडे? समझ में आया आपको? हाँ, 4 PM, ओके ओके। सो 4 PM विल बी ओके उदय लक्ष्मी? ओके, सो ठीक है, विल मीट एट 4 PM। ओके, बाय बाय, टेक केयर।